Respiration in plants, Part 2. Aerobic respiration. Within the mitochondria, for aerobic respiration to take place, the final product of glycolysis, pyruvate, is transported into the mitochondria from the cytoplasm. In aerobic respiration, the crucial events are by the stepwise removal of all the hydrogen atoms, the complete oxidation of pyruvate, leaving 3 molecules of carbon dioxide. As part of the hydrogen atoms to molecular oxygen, the passing on of the electrons removed with simultaneous synthesis of ATP. What is interesting to note is that in the matrix of the mitochondria, the first process takes place while on the inner membrane of the mitochondria the second process is located. By the glycolytic catabolism of carbohydrates, pyruvate, which is formed in the cytosol, undergoes oxidative decarboxylation after it enters mitochondrial matrix by a complex set of reactions catalyzed by pyruvic dehydrogenase, including an AD ion and coenzyme A. The reactions catalyzed by pyruvic dehydrogenase require the participation of several coenzymes. During this process, from the metabolism of 2 molecules of pyruvic acid, which are produced from one glucose molecule during glycolysis, 2 molecules of NADH are produced. The acetyl-CoA then enters a cyclic pathway, tricarboxylic acid cycle, more commonly called as Krebs cycle after the scientist who first elucidated it, Hans Krebs. Tricarboxylic acid cycle. To yield citric acid, the TCA cycle starts with the condensation of acetyl group with oxaloacetic acid or OAA and water. By the enzyme citrate synthase, the reaction is catalyzed and a molecule of CoA is released. Then citrate is isomerized to isocitrate, which is followed by 2 successive steps of decarboxylation leading to the formation of alpha-ketoglutarate acid and succinyl and then succinyl-CoA. Succinyl-CoA is oxidized to OAA in the remaining steps of citric acid cycle, allowing the cycle to continue. A molecule of GTP is synthesized during the conversion of succinyl-CoA to succinic acid. This is a substrate-level phosphorylation. GTP is converted to GDP in a coupled reaction with the simultaneous synthesis of ATP from ADP, where NAD ion is reduced to NADH plus hydrogen ion. There are 3 points in the cycle and 1 point where FAD ion is reduced to FADH2. The continued acetic acid oxidation via this TCA cycle requires the continued replenishment of oxaloacetic acid, the first member of the cycle. In addition, from NADH to FADH2, it also requires regeneration of NAD ion and FAD ion respectively. For this phase of respiration, the summary equation may be written as follows. We have still now seen that to release carbon dioxide and 8 molecules of NADH and hydrogen ion, glucose has been broken down. Besides just 2 molecules of ATP, 2 of FADH2 have been synthesized. Neither oxygen has come into the picture nor the promised large number of ATP has yet been synthesized. You may be wondering why we have been discussing respiration at all. Also, what is the role of the NADH plus a hydrogen ion and FADH2 that is synthesized? Let us now understand how ATP is synthesized and the role of oxygen in respiration. Electron Transport System or ETS, and oxidative phosphorylation. The following steps in the respiratory process are to release and utilize the energy stored in NADH plus hydrogen ion and FADH2. When they are oxidized through the electron transport system, this is accomplished and the electrons are passed on to O2, resulting in the formation of water. Electron transport system is the metabolic pathway through which the electron passes from one carrier to another, and it is present in the inner mitochondrial membrane. During citric acid cycle, electrons from NADH produced in the mitochondrial matrix are oxidized by NADH dehydrogenase, and electrons are then transferred to ubiquinone located within the inner membrane. Also, ubiquinone receives reducing equivalents via FADH2 that is generated in the citric acid cycle during oxidation of succinate. With the transfer of electrons to cytochrome C via cytochrome BC1 complex, the reduced ubiquinone is then oxidized. Attached to the outer surface of the inner membrane is a small protein, cytochrome C, which acts as a mobile carrier for transfer of electrons between complex 3 and 4. Complex 4 refers to cytochrome C oxidase complex containing two copper centers and cytochromes A and A3. In the electron transport chain, when the electrons pass from one carrier to another via complex 1 to 4, they are coupled to ATP synthase for the production of ATP from ADP, an inorganic phosphate. The number of ATP molecules synthesized depends on the nature of the electron donor. 3 molecules of ATP are formed by the oxidation of one molecule of NADH, while 2 molecules of ATP are produced from one molecule of FADH2. Although only in the presence of oxygen the aerobic respiration takes place, and the role of oxygen is limited to the terminal stage of the process. Yet the presence of oxygen is vital, since by removing hydrogen from the system it drives the whole process. Oxygen acts as a final hydrogen acceptor. Unlike photophosphorylation, where for the production of proton gradient required for phosphorylation, it is the light energy that is utilized in respiration for the same process, it is the energy of oxidation reduction utilized. The process is called oxidative phosphorylation for this reason. The mechanism of membrane-linked ATP synthesis, as explained by chemiosmotic hypothesis, you have already studied in the earlier chapter. As mentioned earlier, during the electron transport system, the energy released is utilized in synthesizing ATP with the help of ATP synthase. The two major components F1 and F0 are present in this complex. The site for synthesis of ATP from ADP and inorganic phosphate is present in the headpiece of F1, which is a peripheral membrane protein complex. An integral membrane protein complex F0 forms the channel through which protons cross the inner membrane. Through the channel, the passage of protons is coupled to the catalytic site of the F1 component for the production of ATP. 2 hydrogen ions passes through F0 for each ATP produced from the intermembrane space to the matrix down the electrochemical proton gradient. The respiratory balance sheet. To make calculation of the net gain of ATP for every glucose molecule oxidized, it is possible, but this can remain only a theoretical exercise in reality. These calculations can be made only on certain assumptions that, with one substrate forming the next, there is a sequential orderly pathway functioning, and with glycolysis TCA cycle and ETS pathway following one after another. In glycolysis, the NADH synthesized is transferred into the mitochondria and undergoes oxidative phosphorylation. In the pathway, none of the intermediates are utilized to synthesize any other compound. Only glucose is being respired at any of the intermediary stages. No other alternative substrates are entering in the pathway. But in a living system, this kind of assumptions are not really valid. Simultaneously, all pathways work and do not take place one after another. As and when necessary substrates enter the pathways and are withdrawn from it, as and when needed ATP is utilized. By multiple means, enzymatic rates are controlled. Yet to do this exercise, it is useful to appreciate the beauty and efficiency of the living system in extraction and storing energy. Hence, during aerobic respiration of one molecule of glucose, there can be a net gain of 36 ATP molecules. Now let us compare fermentation and aerobic respiration. Only a partial breakdown of glucose accounts for fermentation, whereas it is completely degraded to carbon dioxide and water in aerobic respiration. There is a net gain of only 2 molecules of ATP in fermentation for each molecule of glucose degraded to pyruvic acid, whereas under aerobic conditions many more molecules of ATP are generated. In fermentation, NADH is oxidized to NAD ion rather slowly. However, in case of aerobic respiration, the reaction is very vigorous. Amphibolic pathway. For respiration, glucose is the favored substrate. Before they are used for respiration, all carbohydrates are usually first converted into glucose. As has been mentioned earlier, other substrates can also be respired, but then at the first step they do not enter the respiratory pathway. First into glycerol and fatty acid, fats would need to be broken down. The fatty acids would first be degraded to acetyl-CoA and enter the pathway if they were to be respired. After being converted to PGAL, glycerol would enter the pathway. By proteases, the proteins would be degraded, and after deamination, the individual amino acids, depending on their structure, at some stage would enter the pathway within the Krebs cycle or even as pyruvate or acetyl-CoA. Since breakdown of substrate takes place during respiration, the respiratory process has traditionally been considered a catabolic process and the respiratory pathway as a catabolic pathway. We have discussed above at which points different substrates in the respiratory pathway would enter if they were to be respired and used to derive energy. What is important to recognize is that it is these very compounds that would be withdrawn for the synthesis of the said substrates from the respiratory pathway. Hence, fatty acids would be broken down to acetyl-CoA when it is used as a substrate before entering the respiratory pathway. But acetyl-CoA would be withdrawn when the organism needs to synthesize fatty acid from the respiratory pathway. Hence, both during breakdown, the synthesis of fatty acids, the respiratory pathway comes into the picture. Similarly, respiratory intermediates form the link during breakdown and synthesis of protein too. Within the living organism, breaking down process is catabolism and synthesis is anabolism. It would hence be better to consider the respiratory pathway as an amphibolic pathway rather than as a catabolic one, because the respiratory pathway is involved in both anabolism and catabolism. Respiratory quotient. Let us now look at another aspect of respiration. As you know, oxygen is consumed and carbon dioxide is released during aerobic respiration. In respiration, the ratio of the volume of carbon dioxide evolved to the volume of oxygen consumed is called the respiratory quotient or respiratory ratio. During respiration, the respiratory quotient depends upon the type of respiratory substrate used. The RQ will be 1 when carbohydrates are used as substrate and are completely oxidized, because carbon dioxide and oxygen are evolved and consumed in equal amounts respectively, as shown in the equation below. The RQ is less than 1 when fats are used in respiration. Calculations for a fatty acid, tripalmitin, if used as substrate is shown. The ratio would be about 0.9 when proteins are respiratory substrates. In living organisms, what is important to recognize is that respiratory substances are often more than one. As respiratory substrates, pure proteins or fats are never used. Thank you so much for watching. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content. For more videos, please check the description box down below.